Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about Actinomycetes. Okay, so at first Actinomycetes is very controversial group because many of scientists thought it as a fungus, but actually it is a bacteria. Okay, so these are gram-positive bacterium, filamentous, heterotrophic, and heterotrophic means they depend on other for their nutrition. Saprophytic in nature, which play a significant role in degradation of organic matter. As they depend on the dead decaying matter, they degrade those matter from the environment and ultimately clean the environment. They have high guanine and cytosine content. They show filamentous structure which can be branched to form stable mycelium. The mycelium is not always intact or stable. They may break into rod or cocci-shaped fragments. When grown, the Actinomycetes branch profusely, forming network of mycelia both on the surface of agar and underneath as well. Surface mycelia growing on substrate is called aerial mycelia, and mycelia submerged beneath the surface substrate is called substrate mycelia. So on the agar plate means on the culture plate containing agar medium, the Actinomycetes grow on the surface and below the surface of the agar, and surface mycelia which grow on the surface is called aerial mycelia, and mycelia submerged beneath the substrate under the substrate called substrate mycelia. Filamentous Actinomycetes are known to produce 2/3 of all known antibiotics which fall under the categories including antifungal. They can produce antitumorous as well as immunosuppressive agent. So these are economic importance of filamentous Actinomycetes. Next, the name Actinomycetes was derived from Greek word atkis and mycis. Atkis means a ray and mycis means fungus. This suggests their structural similarities with fungi, but they are biochemically more related to bacteria. Hence, they are included under the domain bacteria. So they look like fungus, but they have many structural features like bacteria. They have peptidoglycan cell wall. So they are included under the bacteria domain. They are connecting link between fungus and bacterium. So finally it is called as connecting link or bridge between fungus and bacterium. They are also referred to as actinobacteria or actinomycetota. Okay. Next, Actinomycetes are predominantly free-living microorganisms found on a variety of environment. The soil is the most important reservoir for Actinomycetes. Actinomycetes are found predominantly on the soil, and also the soil represents the most active zone of interaction between Actinomycetes and other life forms. And Streptomyces, which is a genus of Actinomycetes. These Streptomyces are significant population of Actinomycetes among all other soil-dwelling Actinomycetes species. The genus Streptomycetes are undisputed genus isolated in every study, which are extraordinarily significant in synthesis of antibiotics. Okay. Then the classification, as I have already told you that these Actinomycetes are included under bacteria domain and phylum Actinobacteria. According to Borges Manual of Systematic Bacteriology, the phylum Actinobacteria is classified into 6 classes. One is Actinobacteria. Now it is called as Actinomycetia. Then Acidimicrobia, then Coriobateria, then Nitrileuruptoria, then Rubrobacteria, then Thermoleophilia. So these are 6 classes included under Actinobacteria phylum. Okay. Next, the habitat and ecology. So these actinobacteria are ubiquitous in nature with high versatility in nutrient requirement, biotic and abiotic interaction. So they are found in almost all type of habitat. They are found in the terrestrial as well as aquatic habitat. Aquatic habitat include both marine and freshwater ecosystem. Several species are extremophiles. They are found in freezing condition called as psychrophile. They are also found in normal temperature called as mesophile, as well as very high temperature called as thermophile. Corynebacterium, Modestobacter, then Streptovorticillium species are psychrophilic species, that means found in very low temperature, whereas Thermoactinomyces, Thermomonospora, Saccharopolyspora, these are thermophilic species. Likewise, some species are found in very acidic condition. Some species are haloalcophilic condition. Some are found in halo means highly saline, and alcophilic means alkali condition, in basic condition. Several species are found in the soil with high salt condition. Some are also endophytic actinobacteria, that means live in the body of the host organism. And numerous genera are also symbiotic. They show symbiosis with plant, animal, and several other organisms. Okay. Then morphology, external features. The diverse morphological feature is seen among the members of actinobacteria. They display different cellular arrangements and branching. In general, they are rod-shaped, bacilli-shaped, and about 0.5 to 2.5 micrometer, sometimes even up to 5 micrometer in length. Some like micrococcus are cocci shaped also. So there are some exceptions. Many species produce hyphal or mycelial growth. These mycelia are also of different shapes. Some are branched, some are twisted, some are straight, some are septated or aseptate. What is septate? Suppose it is a mycelium. Some have cross wall between the cells. These are called as septate. But some lack those septa, are called aseptate. Okay. Next. So aerial mycelium, which grow on the surface bearing spore or spore forming structures. These are key feature in identifying several species. We can identify several species of Actinomycetes by simply observing their spores, pattern of spore formation. These aerial mycelia may be straight or flexus or fascicled or monoverticillate without spirals, monoverticillate with spirals. Some open loops, some so open loops, some so open spirals, closed spirals, biverticillate with or without spirals. So these are different forms of aerial mycelia. 3 different types of spores are mainly observed. One is conidiospore, one is sporangiospore, and oidiospore. Sometimes arthrospores also are formed. Next, few species contains fimbriae. What is fimbriae? These are hair-like projections on the surface of bacteria, and while several lack them, mostly Actinomycetes lack any kind of locomotory organs. So these are mostly non-motile in nature. On culture, when we culture them, most of them show dry pigmented colonies with aerial and substrate hyphae growing, giving a cottony or velvety or powdery appearance like fungus. Pigmentation is seen both in aerial and substrate hyphae. Few can also produce moist colonies because they have capsulated species. Capsule, what is present in your bacteria, glycocalyx. So like bacteria, obviously these are included under bacteria. So like bacteria, some Actinomycetes species also form capsule. Then cell wall composition. The composition of cell wall in Actinomycetes varies greatly among different groups and is of considerable taxonomic significance. Four major cell wall types are distinguished among these filamentous bacteria on the basis of the three features of peptidoglycan composition and structure. So these features are diaminopamelic acid isomer on tripeptide side chain in position 3, the presence of glycine in interpeptide bridge, and sugar content of peptidoglycan. So these are distinguished features of this filamentous bacteria. As is evident in the next table, the table present in the next slide, characteristic sugar pattern is present in three types of Actinomycetes, of those Actinomycetes with meso diaminopamelic acid. So let's have a look on this table. So cell wall type, four types of cell wall is found among the groups of Actinomycetes. Among those, they have diaminopamelic acid and glycine in interpeptide bridge. Characteristic sugar is absent in one type of cell wall, but other three it is present. Mainly consist of aravinose, xylose, medurose, aravinose, galactose, and representative, the name of representative genera are also mentioned here. Next, reproduction. Actinomycetes, like other bacteria, reproduce only by asexual method. The asexual mode of reproduction is accomplished by arthrospore formation or conidiospore formation mostly. In this respect, however, they resemble fungi to some extent, because bacteria mostly reproduce by binary fission mostly, and fungi mostly reproduce by these spores, and Actinomycetes reproduce by means of these spores. So in this field they resemble fungi. Okay. Arthrospores are formed in bacteria having fungus like filamentous body, and generally in Actinomycetes members, and filamentous bodies of this bacteria break into rod shaped smaller fragments called arthrospores, which are capable of growing into new filaments. So this is the Actinomycetes filament. They break into smaller fragments, and individual fragment capable to grow as a separate individual fragments, and these are called as arthrospores. Actinomyces genus show this type of reproduction. Next, conidia formation. It is a common method of reproduction in some members of Actinomycetes. These filamentous branched bacteria produce smaller, oval or rounded structure called as conidia terminally on a certain apical branches called as conidiophore, as in fungi. Each conidium germinates, giving rise to bacterial cell. So these are filaments, and on the terminal part of the filament they produce conidia on a special branch called conidiophore. Okay, and these conidia after detachment form as a new filament, grow as a new filament. Okay, next, so this is the structure of arthrospore and conidia. Okay, so with this I am ending this lecture video. I hope you will like it, and if you like it, please hit the like button and share with your other friends as well as your teachers as well as your family members, and please appreciate my hard work and subscribe to my channel Sunanda's tutorial. Thank you.